Hello, it's a pleasure to be here with you this, well, it's evening here, halfway around the world. And I want to talk to you about Alzheimer's disease, which is a high prevalence sporadic disease that's a major variable in the quality of human life during aging. It's defined on the basis of pathology. So on the next slide, I show you the current definition of Alzheimer's disease is based on so-called ATN criteria. A for amyloid, shown in red here in this two-channel confocal microscopy image, is an extracellular accumulation of a protein called A beta. T is for tau. That is a protein that is accumulating inside of neurons in Alzheimer's disease, shown here in the green channel. It's an intracellular accumulation. N stands for neurodegeneration. These pathological changes are accompanied by the loss of neurons, the degeneration of processes, the inability of cells to communicate with each other. Today, I want to talk about the tau pathology in molecular detail. Now, the tau pathology has emerged as a major staging system for Alzheimer's disease, depicted on this slide in stages 1 through 6, much like cancer biology. It's a geographical concept. The pathology, this T pathology, tau pathology, is beginning in the deep temporal lobe, located here in stage 1, then spreads into the hippocampal formation here in deep temporal lobe, shown clearly here in stage 3, and from there to involve other areas of the brain. By late stage Alzheimer's, it's affecting many areas of the neocortex. This is region here on the surface of the brain, including areas of primary sensory and primary function, such as Heschel's gyrus here for hearing, and Brodmann area 17 involved in vision. So by late stage disease, the pathology is widespread, but this propagation of the pathology from one region of the brain to another is sufficiently reproducible in case to case that this is the major staging system that's used in Alzheimer's disease. Let's take a closer look at what this pathology is composed of, starting here in the superior temporal gyrus. This is what that might look like in 1.5 Tesla MRI. This is a coronal section of the brain. This is what that would look like with an immunohistochemistry that's using an antibody to stain for the tau pathology. The tau lesions themselves, when they accumulate in a cell body, are called a neurofibrillary tangle. The entire cell body is filling with this material. It's spreading into the apical dendrite. And this material, if we look at high magnification, we can actually break these cells open, isolate the material from brain. The tau has a filamentous appearance. These are filamentous aggregates of tau protein that are accumulating in these lesions of disease. And so this has become part of the definition of Alzheimer's disease. A big breakthrough of the past 5 years is to develop the methods to be able to determine the atomic structure of these aggregates and how they vary among different diseases. Again, the protein itself is tau protein. That's a cartoon here in panel A, showing it as about a 400 amino acid polypeptide. It's an intrinsically disordered protein. It means it doesn't have much secondary or higher order structure associated with it. And that's why in panel B, it's illustrated as a coil of polypeptide. In disease, however, this region here near the C-terminus of the protein is adopting beta sheet structure and aggregating to form these filaments. The structure of these, determined in just 5 years ago, is shown here on the right side. 2 protofilaments come together to form a full filament, and all of the polypeptide chains are in parallel in register beta sheet conformation. And it turns out that Alzheimer's disease, it's the leading form of dementia. It's the leading form of tauopathy, that is diseases, neurodegenerative diseases that are accompanied by the aggregation of tau protein, but it's actually one of several. So Alzheimer's disease is shown here on the right. The Other diseases here, especially these called frontotemporal degeneration diseases or frontotemporal lobar degeneration diseases, also accumulate these tau aggregates, but notice that the conformation of each is different. We call this polymorphism. Each of these is a polymorph, or also can be referred to as a conformer. So the research focus, our focus, is on trying to understand this process. We want to know how aggregates form. Each tau molecule has two sequences in it, hexamers that are absolutely essential for aggregation. So they are necessary, but they're not sufficient to dictate this polymorphism that's seen in each neurodegenerative disease, including Alzheimer's disease. So we'd like to know the function of the amyloidogenic sequences. We'd like to understand the precise aggregation permissible pathological cellular conditions that combine with these amyloidogenic sequences to drive polymorphism and lead to the formation of these aggregates that I showed you have been determined at high resolution. We'd like to know how these aggregates spread, as I showed you, from one region to another in a stereotypic fashion. The prion hypothesis is very popular for that, but demonstrating that on a molecular level has not been done yet. That is, in a prion-like spreading mechanism, the conformation would be replicated as the pathology spreads to different brain regions. So we're interested in that problem as well. We'd like to know which conformers are biologically active, these large polymers that I'm showing you, or smaller aggregates called oligomers that also are formed in disease. We'd like to be able to detect individual polymorphisms in living cases, and that's shown here on the right, using chemistry, the development of small molecule probes that could associate selectively with individual folding patterns or conformers. And the long-term goal of the field is to identify methods to slow or halt the progression of this pathological process, because it correlates with neurodegeneration and appears to have a close relationship to it. So I want to tell you a story about how I'm going to focus in on how aggregates form and modeling that in an experimental system. We've pioneered the use of small molecules to drive the aggregation of tau protein in vitro. Normally, tau is an intrinsically disordered molecule, very hydrophilic, has a below average composition of hydrophobic residues. It's very soluble in solution and will not aggregate even at supraphysiological levels. So this requires the addition of exogenous agents to induce the aggregation reaction. And as I said, we pioneered the use of these small molecules. They're dye-like molecules that have a planar heterocycle structure. This one is called Geranine G. It's composed of a naphthalene unit here on the left, connected by a phenyl bridge to a benzothiazole moiety. All of this is extensively conjugated, giving the molecule a red color in solution. Now, this panel C is showing you that the material is available as a highly purified form. This is by high-performance liquid chromatography analysis, by mass spectrometry. It's an individual single species. And again, on the basis of its absorbance profile, it absorbs light in this visible region, so it's nice and red. This is a quantum calculation of the structure of the molecule. The minimal energy structure is planar, with the bonds highly conjugated. It has a strong charge gradient associated with it, drawn from the center of mass of the molecule as a charged molecule. So although it contains two negative charges, that is spread out over the length of this molecule. It has a character of a super chaotrope. So although negatively charged, it's fairly large in size. Now, what happens when a molecule like this contacts tau protein? We compare that to another inducer. We can also do this with anionic micelles, whether that's detergents or vesicles made of lipids. And this is what AlphaFold thinks that intrinsically disordered tau protein looks like, as you see an extended polypeptide chain. When Geranine G is in contact with tau, it induces the formation of these aggregates that are easily detected by electron microscopy. And the unusual characteristic is that the dye remains associated with the tau aggregates after aggregation. So normally we would see a pellet. If we spin this down at high G-force, that would be opalescent or white in nature. When you aggregate in the presence of Geranine G, it remains bright red. And because of the known extinction coefficient, we're able to estimate the mole per mole stoichiometry of about 8 moles per mole remaining associated with the aggregates. So I want to tell you about three-dimensional structure of these aggregates determined by Dmitry Molishka, an MD PhD student in the laboratory, using cryo-electron microscopy, technique that's allows us to determine three-dimensional structure without labels or crystallization of the sample. So this is the structure that he's determined, beginning here in the lower right hand corner with residue 273. An atomic model is here on the right. It consists of a loop structure. It's coated by B factor, which gives you an idea of the flexibility. The most rigid regions of the molecule are shown in blue. The most flexible are shown in red. And the model captures inducers in the act. We can see a series of bound states that are labeled here P1 through P6. P stands for pendant. And there's a major site here that is in parallel orientation called A1. I want to show you what this looks like. If we take a look first at a characteristic pendant site, we can see the molecule. This is about 3.3 angstrom resolution. Every time we see a molecule, almost every time it's associated with a lysine side chain, a positively charged side chain, presumably interacting through sulfonate groups on the Geranine G molecule, always with an intervening density that we interpret as water, consistent with the superchaotropic activity of these molecules. This is shown on the left side in panel A, looking down the length of the filament. Panel B is showing it from the side. And from the side, you can see that these molecules are predicted to form a stack of ligands. They're planar and can associate face to face. This is called an H aggregate, H for hypsochromic. The angle is such that that defines H. So these we have as many as 6 sites that are associated by this kind of a mechanism through lysine side chains primarily and extending out into solvent. The parallel site is shown here. At this level of resolution, we can't fit the molecule exactly, but it does show the tripartite nature of it. We model this density as being the naphthalene. Phenyl is clearly in the center, and the benzothiazole moiety here in this region because it makes contact with this nice hydrophobic region. Now, this one also is associated with lysine residues with water molecules associated with it. What's interesting about this site is that it's perpendicular to the pendant sites. It's coordinating these 2 lysines that are adjacent to one of these nucleation sequences that we're interested in. Panel B shows this from the side where the green chain where the ligand is binding here is associating with this nucleation sequence with a structure that resembles Uzawa-like nucleation structure that he postulated for actin. So we're interested in which of these poses is important for inducing the aggregation of tau protein. That first structure with the H aggregates, it's possible to disrupt that by using non-ionic detergents. This is classically used in the pharmaceutical industry in high throughput screening to avoid the self-association of ligands, which typically occurs at higher concentrations, above 10 micromolar. Many drug-like molecules will self-associate in a face-to-face fashion or even form micelles at a high enough concentration. So this is a sedimentation assay where lanes 1, 2, and 3 are the soluble fraction, and 4, 5, and 6 are triplicates of the pellet fraction. And the 1 through 6 is in the absence of Triton X100 at 0.01%. This is a classic concentration that's used in high throughput screening. And on the presence on the right-hand side. The summary, the integration of this data is shown in panel B, that in the absence of Triton X100, most of the tau is driven into the pellet form in the presence of inducer, and that the presence of Triton X100 does not interfere with that process. So we don't think that the pendant sites, or at least the maybe the charge neutralization is important, but the H aggregate formation of ligand is not necessarily contributing to aggregation propensity. To test which sites were important for that, we did the sedimentation assay after mutagenesis of individual residues. So on the left here is the wild type protein. We can destroy aggregation propensity completely by interfering with those two nucleation sequences that I showed you previously. The y-axis here is aggregation as percent soluble. So when you have wild type protein, there is virtually no soluble material. It's almost all driven into the aggregate form. Whereas if one destroys the ability to nucleate, almost all of it is in the soluble form. So as we inhibit aggregation propensity, we'll see a higher signal on a plot like this. As we mutate the individual residues so that they can no longer bind the Geranine G in a pendant form, we find very little contribution to aggregation until we get to the 317, 321 region. That was the parallel association. And if we map this finally at that one residue resolution, it's the 317 in particular that can account for nearly 50% of the aggregation propensity. When that is converted to a non-charged alanine residue, Geranine G loses approximately half of its efficacy in this system. So we believe that the association of molecules such as this, specific anions that can associate and coordinate with lysines like 317 and 321 in this position, are important for driving the aggregation reaction. And this is important because most of the disease-associated structures that have been described so far have non-proteinaceous densities associated with them, including in this very position here. So how might this work? Again, I note that the molecules associating with a segment that is forming a zipper structure with the nucleation sequence VQIINK located here. So to learn about how this may affect the aggregation reaction, we've turned to steering molecular dynamics simulations. And this is done all in silico. We begin with hexapeptides and a known crystal structure of an isosteric zipper involving one of the nucleation sequences of tau protein. We build up fibrils from that in the computer, and then we energy minimize that structure, and we work with stable things that can hold together under conditions of molecular dynamics. And this is what that looks like here. So these duplexes are stable. And then using steering MD, we're able to pull these chains apart into individual beta sheets, estimate their stability in trajectories, and engage in free energy calculations to identify the energetic sources of stability. Now, to save a little time and also to avoid any kind of computer crash, I'm just showing stills from the molecular dynamics simulation. Normally, these would be moving quite rapidly. On the left is a duplex. This is modeled after Alzheimer's disease, a heterosteric zipper involving a nucleation motif shown in blue and a C-terminal portion shown in red here. This is stable as a duplex throughout the molecular dynamics simulation. If we pull these apart now and look at them as individual beta sheets, we find that the THK LTF, the binding partner, falls apart in a trajectory such as this over 50 nanoseconds, whereas the nucleation sequence, shown here as VQI-VYK, and also we've done it with that VQI-INK sequence, is stable over the entire trajectory. So our model of the activity and then what is driving the aggregation and the formation of these lesions in disease is small molecules that in authentic disease, they could be up to 100 cubic angstroms in volume. You know, these dyes are fairly large compared to that 400 cubic angstroms, with the naphthalene disulfate of warhead being over 200, but it's in the identical position of these densities that are found in authentic disease, that these can stabilize structures for these interaction segments that normally are not stable in beta sheet conformation, and that they can interact with the key nucleating sequences, and that this association is the key event in nucleation. And it's consistent with a nucleus cluster size of 2 that we've determined through kinetic measurements. And again, I draw attention to the close similarity with Ozawa-like structure predicted with actin as the side chains interact. You can see how the association of units could exclude water and help provide an entropic driving force for aggregation. So our conclusions from this kind of work is: how do aggregates form? Non-specific charge neutralization can be part of that. Tau is normally positively charged, and the addition of multiple moles per mole of a negatively anionic species can help neutralize that, minimize the solubility, and that could increase the rate of aggregation. But there's also a specific effect, specific stabilization of a segment that's able to interact with the nucleation motif. And that can not only nucleate the formation of these aggregates and lesion formation, but also have a major effect on the polymorphism that the structures adopt. And so our future directions, we're correlating inducer structure with tau polymorphism. We have a series of inducers that we're looking at, capturing the inducer in complex with the aggregates to get information about their site of interaction and the polymorph that they're capable of stabilizing. And we're also carrying out more steering molecular dynamics simulations of diverse heterosteric zippers, including those that appear in those frontotemporal lobar degeneration diseases, not just Alzheimer's disease. To help support this, we've built a series of Alzheimer's disease resources in the United States, funded by a U24 mechanism at the NIH. So ours on tau protein is called the Interdisciplinary Resource Network on Biologically Active Tau Aggregate Polymorphs from Alzheimer's disease and related dementias. We're preparing aggregates from human brain in large quantities, providing that to the research community for experimentation, including seeding studies. My collaborators on this are at the University of Washington, at University of Texas Medical Branch, and UCLA. So to acknowledge the people who have done this work, especially Dimitri Malishka and Austin Allen, are responsible for the structure that I showed you today. And Jae-Hoon Yang and Sherwin Singer's lab did all the molecular dynamics simulation. Thank you for your attention.